Thank you so very much for those of you who are in the room who came for Kara's talk. I greatly appreciate that, and we're excited for those who are joining on Zoom as well. I want to tell you a little bit about our speaker today before she starts. So Kara actually did her undergraduate at William and Mary, and then she did an internship in a very famous seagrass ecology lab. From there, she went and worked at AGU for a bit before deciding to continue on with grad school. And she then went down to Dolphin Island Sea Lab and was working, has been working in a really very exciting lab led by Kelly Dorgan, looking at sediment. And Kara's interests, which you're going to hear about now, are kind of like the animal involvement in that. Well, Kara reached out to Niels and I, getting towards the end of her PhD, looking for the next step, and was like, are you guys willing to write a proposal, with me? And of course, we're like, sure, absolutely. And so that started a lot of really interesting conversations with the hospital, interesting conversations with other people in other institutions, and Kara put together and she put in, and she actually wrote a successful NSF postdoctoral fellowship, which of course is difficult to do. And so she'll be joining SoMAS in May, or I guess June.
Mayish, June-ish.
Mayish, June-ish, to start the work here. And so I'm grateful for you coming, knowing that Kara's a known commodity. She's coming here. She's going to be spending a couple of years with us doing her postdoc, and she's excited about a lot of things that I think might be very interesting to many of you in the room. So having said that, let's welcome Kara Gadeken.
Almost Dr. Kara Gadeken. Well, thank you so much for having me. I'm really excited to be here. I wanted to take the opportunity today to talk a little about me, introduce myself, but also talk about some of my graduate work and then what I'm planning on doing while I'm here at Stony Brook. So just first, to get the acknowledgements out of the way, everyone in the sediment ecology lab at the Dolphin Island Sea Lab, and faculty and staff that have helped me over the past many years, my funding sources as well. Okay, so outline for today's talk. I'm going to start out with a little bit of personal background. I'm going to give you a marine sediments crash course in case you're unfamiliar with them, talking about my graduate work on temporal variation, particularly in oxygen, and then my postdoc plans on spatial variation in seagrasses. Okay, so I grew up in Alexandria, Virginia. It's this little arrow right there in Northern Virginia. It's a suburb of DC, a big sprawling suburb. So I grew up in a little box made of ticky-tacky, but got out into nature every once in a while, got to go to the beach, but didn't really think all that much about a career in marine science until I got to my undergrad, where I went to William and Mary. I got my bachelor's in biology and a marine science minor. And then after I graduated from undergrad, I went to the American Geophysical Union for a year, and I worked in their education and outreach department. But during my undergrad, I joined Emmett Duffy's seagrass biodiversity lab at the Virginia Institute of Marine Science. I joined that my freshman year. And this was my first experience being out on the water, doing research, and being able to be in the seagrass beds and learning about the ecosystem was just the best thing ever. I had never done anything like it. My favorite days in particular were the monthly field samplings, where you could go out and really see the dynamism of the seagrass environment and how much they change throughout the seasons. So I worked in this lab for all 4 years as a part of the ZEN project, or the Zostra Experimental Network. And I really got a lot of great experience in that lab, working in seagrasses. And I got to go to a bunch of different sites. I started out as a volunteer, but eventually ended up in more of an intern technician role, and I was sent all over the place to the sites in this worldwide network of sites. What was I saying? Oh, yes. Okay, so I started as a volunteer, but then eventually I ended up in more of an intern technician role, and I was sent to all these different ZEN sites. So the local site was in Virginia, but I also got to see seagrass beds in North Carolina, and we went to a site in Massachusetts. and all the way over to Vancouver as well. So I was sent there to help coordinate the samplings there. So I got to see a lot of really interesting differences in how Zostera ecosystems were structured throughout both its geographical range on the East Coast, but also the differences between the East and West Coast. So this was a really formative experience for me. It definitely left an impression about the importance of understanding how ecosystem variation affects the ecosystems both through time and space. Not working.
Push the advance button.
I am pushing the advance button. This is not advancing. Here, wait a minute. There we go. Okay. So after working for a year, I went back to school at the Sediment Ecology Lab at the Dauphin Island Sea Lab on Dauphin Island, Alabama, to work in the Dorgan Lab. So our lab had a wide array of interests that spans disciplines and scales, often using custom-built instrumentation to address novel methods and investigating how sediments function and how particularly animals interact with sediments and their environment. So working in a sediment lab, much of my graduate career was spent like this, hands in the mud, ass in the air, just like every self-respecting sediment scientist. Sediments are complex and weird and cool habitats, and there's a lot that we still have to learn about them. So before jumping into talking about my work, I should probably give a little bit of background into these ecosystems. So, marine sediments crash course, it'll be painless, I promise. So one of the most important things to know about sediments is that they are fundamentally vertically structured. So inorganic stuff like eroded rock grains and bits of shell will wash into the ocean by rivers, particulates from the atmosphere, things like that. And as well as organic stuff like dead plankton, fish poop, detritus, etc., will sink down from the water column and settle onto the ocean floor. So what you end up with is this heterogeneous mixture of particles composed of both inorganic and organic components that are accumulating on the seafloor. So the goop of the organic material forms this sticky matrix between the sediment grains, and there's a complex microbial community that lives on the grains and in between those grain spaces that respires and breaks down the organic material. So this means that sediments fill this functional role in the oceans of kind of a compost pile, right? Where dead stuff settles down to the ocean floor and dissolved oxygen in the overlying water diffuses in. The dead stuff is broken down by the microbes in the sediment via aerobic respiration, which uses up the oxygen. And these sediments release nutrients like carbon and nitrogen and phosphorus in the process. So some amount of this dead stuff is too difficult to break down and eventually gets totally buried or quote unquote locked away. But by and large, sediments are actually really good at this process. So we know that sediments do this, but there's still a lot of unknowns about exactly how they do this. So a lot of sediment research revolves around figuring out new ways to creatively see through mud to observe and quantify what's going on in the sediments without, disturbing them too much. So what we do know, we do have a good idea of the chemical environment in sediments, at least in a steady state condition. So this is an example of a sediment core. So oxygen diffuses into the sediments, right? But only to a certain depth before it's all used up in respiration. So that's this rusty brown color at the top. So there it's oxic, but below that is anoxic mud. And there's some really fascinating chemistry going on in this anoxic layer. So the microbes there can respire molecules other than oxygen, and they'll do so in this cascade of decreasing efficiency that corresponds with depth. So these chemical pathways are really important, important enough that sometimes you can smell them. For example, at a certain depth, microbes will start to perform sulfate reduction, which produces sulfide, which is rotten egg smell, right? So when you're driving by a marsh and you smell something stinky, that's what that smell is. So animals in sediments can disrupt this delicate layering. Animals like worms and clams and burrowing shrimp, and change both the physical and the chemical composition of the sediments by mixing them around and flushing water through them. So this turns it into kind of a complex mosaic of oxic and anoxic regions throughout the sediment, which can be very cool to see, but very difficult to quantify. On top of that, coastal sediments are also perpetually subject to shifting environmental conditions. So this is an example from my home institution. That's Dihyal right there in the little star. And the sediment plume that comes out of Mobile Bay. So here, sediments are constantly being deposited and then eroded and then deposited again. We also in the system see dramatic fluctuations in salinity and temperature and dissolved oxygen, wind and wave action that make this a system that's constantly in flux. Not every coastal system is as ridiculous as this, but the point is that variation is a defining feature of coasts. So sediments are complicated enough on their own where nothing is changing. So it's a pretty formidable challenge to try to understand how they're responding when they're perturbed by things like burrowers or rooted vegetation, or when environmental conditions shift. But it's important because in situ, these systems are not at steady state, right? We need to understand how they respond to change in order to understand how they're really working. So a guiding question for my graduate work, but also for my future work on sediments in general, is how does variability affect sediment ecosystems? Now, this is a huge question that amounts to several entire careers worth of research, and many researchers have already done a lot of foundational work on this, on untangling these relationships, some of whom are sitting in this room or listening in. But the goal for my research and the approach that I take to studying sediments is from this perspective of variation, right? Building a better mechanistic understanding of how sediment ecosystems work by observing their response to change. And the aim is to kind of keep chipping away at this question, starting with my dissertation work. So let's talk a little bit about that. So for my graduate work, I decided to focus on a type of variation that's incredibly important in sediments, dissolved oxygen. So oxygen availability is a driving factor in sediments. It's kind of this nexus around which a whole host of biogeochemical and ecological interactions are revolving. So remember that redox cascade, it's fundamentally dictated by oxygen availability. The burrowing critters change the way that sediments function in large part because of how their activities change the distribution of oxygen in sediments. So I started by looking at how change in oxygen affects the mixing of sediments by fauna. So just as a little bit of background about low DO in general. So low DO can cause some pretty dramatic changes in the sediment environment, both in the sediment itself and the fauna that live in it. So in high DO conditions, we have happy mud. It's this rusty brown color. Remember, that means it's oxygenated. And we have big fauna burrowing around in the sediment, mixing it around, which enhances the oxygenation. However, in low DO conditions, sediments are deoxygenated and the fauna can't survive. They die off and we lose that sediment mixing function. But it's not always as simple as oxygen gets low and everything dies. One of the really interesting things about low oxygen is the continuum of responses that we can see from fauna that may have implications for sediment function. So this is a schematic from Brad et al., who did a survey of animal behavioral responses and the points of mortality specifically as the environment goes anoxic. So here on the x-axis, we have the duration of low oxygen. On the left y-axis, we have the dissolved oxygen level. And on the right y-axis, we have a hydrogen sulfide, remember that toxic chemical. And they devised this schematic. So when DO is very low for a long time, things will tend to die off, right? If it's low for an intermittent amount of time, maybe days or weeks, something like that, we see severe and obvious behavioral responses. But then there's this area of short-term, pretty short-term low oxygen that may be affecting animals sublethally, not killing them off necessarily, but causing changes in their behavior. So there's a kind of short-term low DO that's extremely common in coastal ecosystems, and that's, that is Dihyal oxygen cycle. So this is an example of summertime oxygen cycling in the bottom waters off of Dauphin Island. So we can see this pronounced Dihyal cycle where at night the oxygen gets low because respiration is dominating, right? It's using all the oxygen up. During the day, we have photosynthesis driving oxygen levels to increase. And it can actually get pretty low, pretty close to hypoxic, which is 2 milligrams per liter, and go all the way up to saturated or supersaturated during the day. So when I saw this kind of variability, I decided to focus this part of my dissertation on answering the question, how does mixing sediments by macrofauna change throughout the Dihyal cycle? So I came up with a few hypotheses for what I thought might be happening. So first thing that I thought might be happening was there's this proportional response where the mixing activity of these critters is varying proportionally with the oxygen concentration. Alternatively, you might expect something like what I call a gas response, where when the oxygen is coming back up during the day, the animals kind of overreact to having access to oxygen, kind of like a after the low DO period, and increase their activity proportionally, or not proportionally, increase their activity at the beginning of that increase. So we could also maybe expect something like a lag response where the animals are stressed from the low DO period during the night and take a little while to recover their mixing rates as the oxygen is coming back. So how do we test this? Sorry. So one way is to expose animals to a controlled DIOL cycle in the lab and measure their activity at different points in the exposure. So the problem here is that it's really not easy to manipulate oxygen in the lab. It's actually a major sticking point for studies on oxygen variability. Most of the setups that do it are complicated or expensive or both. So I needed a simple way to do it. So I built one. So this is the oxygen manipulation machine, or OM, very zen. So the way that it works is you have your an experimental tank where you're keeping replicates of little tubs with animals in them. We have a layer of organic rich mud at the bottom of this tank that draws down the DO in the experimental tank. And then we have a reservoir tank with bubbled water. And then there are a set of electronics that are programmed to manipulate the oxygen in the experimental tank by periodically allowing in water from this reservoir tank. And it can drive the oxygen to follow a prescribed pattern in doing so. So I set this system up. Here you can see my Arduino control system. Our Arduino is just a little, basically a mini computer, microprocessor. The experimental tank and the reservoir tank. So the real question is here, does it work? So this is an example of a trial run of the system. So we have the time of the exposure on the X and the DO on the Y. So yes, it works. Sometimes it struggles a little bit to draw down the oxygen. But overall, it does a really good job of manipulating the DO. So I can use this then to expose the animals to a DIOL cycle. And that's what I did. So for this experiment, I chose 3 different taxa. The banded brittle star, which buries its disc a few centimeters down in the sediments, sticks up its arm and use it to feed. We have the polychaete worm, Alinea fusiformis, which is a surface deposit feeder / suspension feeder. It fills this shelly tube that it constructs in kind of shingles, and then it uses its crown to feed on the surface or in the water. And the burrowing clam, Maritella versicolor, which is pretty small on average, usually about 1 centimeter, and can suspension or surface deposit feed as well. I also had a controlled treatment with sediment, but no in fauna. And I chose these 3 taxa because they're all locally available. They're all suspension / surface deposit feeders, but they have some key differences in the physiology and the motility that provides a range of different behaviors to kind of classify their response. So I collected these animals from Pettigoy Pass, which is in between Dauphin Island and Pettigoy Island. So shown here is DO for about a month around when I collected my animals. And we can see that there's a lot of variability, right? So the fauna are naturally experiencing recurring low DO. And I collected the animals and then brought them back to the lab and prepped the experiment. So another big challenge here is to studies like this is how to observe mixing by sediment dwelling animals, right? You're literally trying to see through mud. It can be difficult to observe them non-invasively while also getting meaningful measurements. So for this experiment, I used a couple different methods. So I used luminophores, which are fluorescent sediment particles, to quantify mixing activity. The animals were also kept in these thin aquaria, or what I call ant farms, really they probably should be called worm farms. So I could hopefully observe their activity below the surface. This didn't really end up working out because the fauna are annoying and didn't cooperate. But the luminophores actually ended up being pretty interesting. So I'll show you that. So to prep the experiment, I put my different animals into the ant farms with sediments and as well as the control that had no animals in it. I placed them in the experimental tank and then manipulated the dissolved oxygen in the tank into the DIOL cycle. And then I used luminophores to quantify the mixing activity. So note here, I had a top-down camera with some UV lights in order to show the luminophores. So this is the pattern that the OHM generated. I think it did a pretty good job. So one of the things I noticed was that animals could clear the surface luminophores within a matter of hours. So I had to add more at each of the blue points. And then I took before and after photos at each kind of 6-hour chunk of time, and then comparing them could estimate mixing for each of those periods. So for example, we have the difference between 0.1 and 0.2, where you can see there's a decent amount of mixing, as opposed to comparing 0.17 and 18, where there was comparatively less mixing. So what did I find? I'll just show you the plot of the control treatment first. So we have the same axes. We have time on the X, DO on the left Y, but I've added this axis here, which is the luminophore cover, and that corresponds to these points up here. So note here that further down on the plot means more mixing of luminophores down into the sediment. Just remember that. Okay. So the mixing in the control was low and stable throughout, which is good. We wouldn't expect there to be a ton of mixing in a treatment with that didn't have any animals in it. But for the animal treatments, we did see this kind of distinct pattern come up. So I'm going to take the control away and just show you the data. So the first thing we notice here is that all of these patterns are following a similar general pattern, where they start with high mixing and decrease with kind of a punctuated change throughout the exposure. But remember that our question, right, was about the response to the DIOL cycle, that proportional gas lag alternative hypotheses. So when we test that, we see that the only irregular response that we see is in the first DIOL cycle for the brittle star over here, where it's kind of doing a Gatsby thing a little bit. That was the only significant difference. But in the second DIOL cycle, we didn't see the same effect. So generally, it looks like there's this proportional response going on. But that is not the whole story, right? Because we still have this overall pattern. So when we look then at the overall pattern that we see, it kind of looks like the combination of two different responses, which is the variation over the DIOL cycle, but also kind of what you can describe as acclimation across the DIOL cycles, like throughout the exposure. So we can actually write an equation and fit it to the data to describe this pattern. And then once we have this equation, we can calculate the confidence intervals around each of these coefficients. And the term is significant if the confidence intervals do not include 0. So when we did that, we found that none of the confidence intervals included 0 except for the one for the variation term in the brittle star, which means that it's probably better described by this power function, right? By this acclimation term. So what this means is that, well, first of all, let's go back to our hypotheses. Does sediment mixing follow the proportional response? Yes, but also no, which is my favorite answer to a yes or no question. Overall, we see that fauna are responding proportionally to DIOL oxygen, but they also have this pattern of change over multiple DIOL cycles. So what this means is that DIOL cycling may cause coastal fauna to mix sediments in situ at lower rates than we might think. So most lab studies on sediment mixing by fauna are done in consistently high dissolved oxygen levels. Or when they do incorporate low DO, it's usually as a kind of exposure where you start with high DO and then you decrease gradually, and then you see what they do with low DO. So this study here is showing that the short-term varying DO, which is the kind of DO conditions that these animals would be consistently experiencing, has unique consequences for how animals are interacting with sediments, notably their mixing. So for my second part of my dissertation work, I kind of shifted focus to the effect of DIOL oxygen cycling on sediment metabolism, which is a critical ecosystem function. It's basically kind of a measure of the compostiness of sediments. So if we go back to our original schema of what happens in low DO conditions, we can look at it from a different perspective. So This pattern means that the change in DO that we see here means that in high DO conditions, we have high SOD or sediment oxygen demand, which can serve as a useful proxy for metabolism. But in low DO, so sediment oxygen demand drops way off because there's no oxygen available, and the animals that are helping to mix the sediments to oxygenate it are all dead. But remember that the DIOL cycle could be having these unique effects. So the sublethal effects of DIOL low DO could be changing faunal behavior in interesting ways that are different from more severe or persistent low DO, and therefore changing the effect that the animals are having on SOD. So my question for this part of my dissertation was, are sediment metabolism rates throughout the DIOL cycle affected by changes in faunal behavior? So if we look at a possible hypothesis for this, we can see that in low fauna conditions, we might expect that during the day, during high DO, we would have high SOD, and then it would drop off at night when the oxygen gets low, and then increase again during the day. In high fauna conditions, we might expect that SOD would be much higher during the day due to faunal activity, plenty of oxygen around, they're pumping, irrigating. It would drop off at night as the oxygen decreases and the animals are stressed, and then increase again during the day. So this basically equates to the proportional response hypothesis from the lab experiment. The question is, does this hold for sediment metabolism? So here, even though I'm interested in the effect of temporal variation, there is a spatial element to this study, in that I needed to connect the temporal variation to in SOD to specific fauna residing in a patch of sediment. So why hasn't this been done? Well, it's actually really difficult to measure SOD, particularly in situ. There are a couple of ways that people have typically done it. The long used, tried and true method is the batch style benthic chambers, where you have basically a cylinder that you chunk into the sediment and cover it, and then observe the change in a given chemical of interest through time. So this is useful because it isolates a patch of sediment, so it has very high spatial resolution, but the temporal resolution on this method is pretty low. It's typically only deployed once or maybe a couple times a day. A method that's has more recently been adapted for measuring sediment flux is the eddy correlation technique, which calculates flux from the flow field above the sediment surface. So this method has very high temporal resolution. You can usually get measurements on the order of minutes, but pretty low spatial resolution, as it's most appropriately used or understood as a spatially averaged measurement technique. So for my experiment, I needed a method with both high temporal and high spatial resolution. So I did what I do, and I built one. So this is my flow-through chamber system. We have it comes in two major components, where we have a central housing that has the electronics and the power for the system, and then that controls several replicate benthic chambers. And I know that this central housing does look a lot like a bomb, but it says right on it that it's not a bomb, so it's fine, don't worry about it. So the way that this works is that the system is deployed as a whole. Each of the chambers has an oxygen probe mounted to the top, sensing the oxygen in the overlying water of the chamber. And then it will allow the system to sit for about 20 minutes as the oxygen in the overlying water is declining. And then after about 20 minutes, it will flush the overlying water with water from the ambient environment of ambient dissolved oxygen. I also put a mixing doodad on the top of each chamber to mix the overlying water and to prevent stagnation. So theoretically, the results from this, or the oxygen patterns that we should see from this, should look something like this, kind of a sawtooth pattern, right? Where we have, okay, so time is on the X, dissolved oxygen is on the Y, and the ambient dissolved oxygen of just the environment is in the blue, and the DO in the chamber is in the black. So here would be that 20-minute incubation where the system is sitting and the oxygen is decreasing. After about 20 minutes, the pump turns on, bumps the oxygen back up to ambient levels, and then starts another incubation. So we can take the slope of that, each of those teeth as a measurement of SOD. and do this repeatedly throughout the DIOL cycle in order to compare SOD at different times. So for this experiment, I picked a site close to home, Bon Secours Bay in Mobile Bay, Alabama. It's a shallow, pretty sandy site with easy shore access. I did 3 24-hour deployments, and also the chambers were covered to isolate the effect of respiration and exclude photosynthesis. And I took measurements of the chamber DO for each of those replicate chambers, as well as the ambient DO, and I collected the faunal communities in each chamber after each deployment. So there are a few questions that we need to answer about the chamber system before we can even look at the SOD data. So the first is, does the ambient DO follow a DIOL cycle? So this is the ambient DO data from each of the 3 deployments overlaying on each other, time on the X, DO on the Y. And the gray box is the nighttime hours. So we can see that it does a pretty good job following the DIOL cycle in all 3. So the ambient DO is following this DIOL cycle. So then the next question is, does the chamber measure SOD, aka does it sawtooth? So this is, I've added the data from one of the chambers, one of the replicate chambers in the black. All the vertical lines are just denoting those 20-minute incubation periods. And we can very clearly see this sawtooth pattern. And also the oxygen at each of the peaks is matching the ambient pretty well. So we can reasonably associate each of these measurements with a given ambient DO in the DIOL cycle. So then question after this is, can we calculate SOD? So here I've added another axis. You can see on the right in the red. And here I want you to know that the SOD axis is actually inverted. So greater SOD, more oxygen going into the sediment, lower on the plot. So yes, we can calculate SOD and then use it to answer the question, does ambient DO drive variation in SOD throughout the DIOL cycle? So these are the data from all 3 deployments. The axes are the same for as the previous plot. And my first thought when I saw this was, that is a huge mess. But most of the values are pretty low and fall in line with what we would expect from this environment. It's a shallow sandy site. Also, this kind of short-term variability is not at all uncommon, especially if we're looking at high frequency eddy, comparing it to the high frequency eddy correlation data, and the fluxes that we see, particularly out of Peter Berg's lab. But also remember that the first step to answering our question is to look at the relationship between DO and SOD. So if our hypothesis holds true and they are proportionally related, we might expect a pattern like this red line, right? So do we see that? No, no, we don't. Not even a little bit. I like to say it looks like buckshot on the side of a barn. So when I saw this data, I thought it would be a good idea to kind of step back and think more about the relationships between the actual parameters that I was interested in. So I did a multiple regression of dissolved oxygen and biomass to see their effect on SOD. I found that DO is not significant, but we could have told that just from this plot. But biomass was significant, and the interaction between DO and biomass was also significant. So there's something going on here between the DO and the fauna, which means we should probably look at the fauna. So this is the fauna data that I got from pulling each of those chambers. We have each of the samples on the X. The abundance is on the top plot, and the biomass of the major constituent fauna is on the bottom. So there's some decent diversity in the samples that I was getting, but by and large, biomass was dominated by just a couple different taxa. So predominantly Nereid and Orbineid polychaete worms, which are burrowing deposit feeders. We also found, let me see if I can move this box. There we go. There we go. Okay.
Click the smallest, yes.
My nails. So there was also this big burrowing wormfish in sample 11, which was super cool to see. So if we were to plot the wet biomass against the sediment oxygen demand, we can see this significant positive relationship between the two of them. So we can even see actually that wormfish and the effect that it's having slightly increasing SOD above what we might expect from that relationship was a really cool example of even one individual having a really outsized effect on SOD. So what this means is that fauna are indeed driving higher SOD. But this alone doesn't explain the interaction with DO. So what's going on there? So to detangle this, I went back to the relationship between DO and SOD, and then just plotted each chamber by itself, that relationship. And if we look at that, we can see this relationship start to emerge. So in low biomass samples, like for example, sample 2, SOD decreases as DO decreases, but in high biomass samples, for example, sample 3, SOD increases as DO decreases. So if we plot these slopes between, or the relationships between DO and SOD against faunal biomass, we can see this significant relationship. We can even see that pesky wormfish again, messing up my regression. So what this means is that fauna are flipping the DO-SOD relationship. We also found that the short-term variability, meaning the point-to-point variability, measurement to measurement, like next to each other, was also greater when fauna were present. I did a bunch of statistical gymnastics to figure this out. You can ask me about it later and I can explain it in agonizing detail. But basically what this means is that in low fauna conditions, this pattern that we expected did actually bear out. It was higher SOD during the day, lower at night, and increased again. And remember, this is what we expected from the high fauna areas, where it was higher during the day, lower at night. But what actually ended up happening was this, where fauna are increasing SOD at night. And actually factoring in the short-term variability, it really looks more like this, where there's a lot of variability on short time scales, overall more SOD during the night due to faunal presence. So what have we learned from this? Well, we know now that the interaction between the DIOL cycle and the faunal behavior is driving both greater and more variable SOD. Fauna are flipping that SOD-DO relationship throughout the DIOL cycle. So this means that we can't really extrapolate daytime or even one-time SOD measurements through time and space, which is very, very common in studying sediments. And that SOD is highly dynamic on short time scales and also dependent on resident fauna. So take-home messages overall from both of these experiments is that there's a short-term DO variability that's driving these unique patterns and faunal effects, both in terms of their sediment mixing, but also their influence on sediment metabolism. And that there's this complex temporal variation in the environmental conditions that may drive as of yet undescribed and unaccounted for changes in faunal effects on sediment function. So while I was finishing up my dissertation, I decided that a good next step would be to kind of shift my focus to better understanding spatial variation in sediments. So I had an idea kind of on simmer on the back burner in my brain since I had started grad school, and having cut my teeth in research working in a seagrass lab and then moving into learning so much about sediments, I naturally started thinking about seagrass sediments. So I wrote a proposal and SF liked it and gave me money. So I'll tell you a little bit about it and what I'll be working on here while I'm at Stony Brook. So a little bit of background. One of the many ecosystem services provided by seagrasses is their exceptional ability to sequester carbon. They only occupy about 0.2% of the ocean's area, but account for at least 10% of the annual blue carbon burial. So the way that they do this is detritus and organic material are collecting at higher rates in seagrasses and are buried with buried with newly deposited sediment and also plant matter. And since seagrass sediments tend to be both less oxygenated and more reducing than unvegetated sediments, this material is remineralized at lower rates and then buried, which quote unquote locks away the carbon. So in the most extreme examples, like for example, this bed of Posidonia oceanica in the Mediterranean, they can form these dense mats of dead and decaying organic material, and you could probably imagine there's a lot of carbon in there. So note here that the ultimate fate of the carbon depends on whether or not the organic matter is being remineralized in seagrass sediments, otherwise known as sediment metabolism, right? So it's really important to understand what is driving seagrass sediment metabolism in order to understand carbon cycling in the ocean. So a little bit more about seagrasses in particular. So seagrasses are anchored in sediments by rhizomes, which are these horizontally extending structures, and roots, which branch from the nodes on the rhizomes. And we know that this structure influences the surrounding sediment, perhaps most notably by leaking oxygen from the roots in order to prevent toxic hydrogen sulfide from invading root tissue. But what this means is that it's generating a chemical gradient with proximity to the root that is creating a unique environment for both the microbes and the macrofauna that are living in seagrass sediments. And yet, all most people do with below ground seagrass biomass is yank the plants up and weigh the roots and rhizomes. I think there's a lot more going on in these sediments than we currently know about. And it's important to understand both the physical structure and also the chemistry around the structure. And in order to kind of understand or resolve the uncertainty around metabolism rates in seagrass sediments. So my question for this project is, how does the seagrass root / rhizosphere influence seagrass sediment function? So the main hypothesis that I have for this project is that the sediment or the seagrass roots are oxygenating the below ground sediment area. And the more volume oxygenated, we can connect that to more metabolism. So these geochemical gradients that are radiating from the roots are oxygenating the sediment in these little halos, more halos, and the more overlap in the oxygenated volume, the more metabolism we might expect. I have several sub-hypotheses underneath this main hypothesis, but this is the core of what I'm trying to figure out. So how do we do this? Well, we can get measurements of metabolism using my fancy benthic chamber setup, but how are we supposed to describe the below ground structure? So we run into here the same problem that sediment researchers have always had, which is how do you see through mud? Well, it turns out that we actually kind of do have some ways to see through mud now. It's more of a matter of figuring out how to weave these different methods together. So the first problem that we have is how to map the physical structure of the seagrass roots and rhizomes. So there's been some pioneering work using MRI in order to map faunal burrows, but it hasn't ever, at least to my knowledge, been used to map seagrass sediments. So we're working with some collaborators at Stony Brook Medicine to put cores full of mud in their $1 million machines and to scan them. And they've actually already tried out scanning the core. This is a still image of a side view of the core. And the video here is the sample core. So MRI kind of scans things and slices and stacks the slices together. So this is a top-down view of the core, and we're moving down through the core. So here, all the bright spots that are lighting up are the roots and rhizomes. So if we assemble that scan into a three-dimensional image, you can get something kind of like this. So here, the core is on its side. That disc-shaped chunk on the top is actually the water on top of the core. But you can clearly see all the root and rhizome structures, which is very cool. So the next challenge for this study is how to measure the geochemical halo around the physical structure of the roots. And we have tools to do that too. So we can use remote chemical sensing methods, for example, planar optodes and microelectrodes, to measure that geochemical halo around the roots, and then overlay that onto the physical information provided from the MRI. So planar optodes in particular can give a really good look into subsurface oxygenation. They've actually been used in seagrasses before. That's part of the reason why we know that seagrasses do this halo thing. It can give essentially a 2D heat map of the geochemical concentrations that can then be extrapolated out into three-dimensional space, which is very cool. So the plan here is to learn how to use planar optodes from Niels, who has done this a lot, to measure particularly sediment oxygenation by irrigating its fauna. So overall, the goal of this project is to try and detangle some of the factors that are driving seagrass sediment function by linking the physical and the geochemical complexity of the seagrass sediment environment to broader seagrass ecosystem processes. I'm really excited to get started and to work with you all for the next couple of years. So thank you. I'll take any questions.
really enjoyed it.
Thank you.
And I was particularly excited by the cartoon of the rhizosphere showing those gradients.
I made that cartoon. I'm very proud of it.
I saw it before. I think I want to branch proposals. Anyway, what's known about chemoautotrophic bacteria in the rhizosphere?
Well, so I actually just talked to one of Brad's old students who had looked at chemosynthetic clams in sediments. And there's some very interesting relationships on facilitation between chemosynthesis in sediments and the seagrass itself. As far as I know, not that much is known. At least I don't know that much. So yeah.
We should talk because those gradients look exactly like the systems that I've built my career on, which are anoxic water columns that have those same gradients.
Oh yeah, that's cool.
They're at the interface between where oxygen disappears and sulfide disappears.
Yeah.
Are these large productive zones? And I expect to see the same thing around the rhizosphere.
Yeah, something that we were talking about was bringing someone in to do some microbial work, looking at the microbial.
Gordon, one of the interesting things, right, is that these gradients are switching, right? During the day, you're pumping all this oxygen in, right? And at night, you're sucking it all out. So that gradient's constantly flipping back and forth,
A lot of these chemoautotrophic constitutes nitrogen, so they're
Other questions? You should probably check the Zoom, right?
I have a couple of questions.
Ah.
I'm shocked to be
Yeah, actually, I'd like to ask a question about this, but I want to go back first to your mixing versus DO characterization. And according to your data, the mixing goes to 0 after 2 cycles.
Yeah.
Is that correct?
Yes. Okay. So
So is that real? Do you think that's a artifact of the experimental setup, or do you really think that that's true?
oh no, I don't think it goes to 0. No, I think that's an artifact of the exposure. But I think the decrease is real. Part of what I would have liked to do, that experiment was a monster to run. I would have liked to run it for a little longer to see what happened over multiple DIOL cycles, because I only did 2 because I expected to see a pattern within the DIOL cycle. So I thought, I'll do 2 and then see what happens, but then ended up seeing that pattern. Also at the end of that exposure was a chunk of time that was very low. It was hypoxic for 6 hours. So it didn't go up again at the end. I suspect that might have had something to do with why we saw that pattern. So I do not think that it's real that mixing goes to 0. But I think the overall pattern of decreased mixing with shorter term oxygen variability is real. Yes.
The other question I had concerns how you're defining sediment metabolism.
Yes.
And in these highly variable and spatially and temporally variable systems that may not be at a local steady state, then the SOD doesn't necessarily reflect sediment metabolism. So I'm wondering if you might include, and probably should include, another measurement of metabolism,
Yes.
for example,
CO2. Yeah, we were actually just discussing that recently, how particularly for this experiment, looking at oxygen dynamics in sediments that are populated with plants is fishy, to say the least. That very complicated and difficult to detangle the oxygen dynamics. So I expect if DO was the only thing I was measuring when I put my pores in, that would be very messy. So we were talking about trying to incorporate some measure of DIC as well to kind of reconcile the variability there. Because so SOD is a much more reasonable measurement of metabolism in unvegetated sediments. But as soon as you put plants in there, that gets messy. So yeah, we're definitely considering that.
And I know, just one last comment, because I know questions might come up, is the plants themselves respond to the diurnal cycle in photosynthesis and DO availability. And I know in that pattern, the rhizosphere exports different anaerobic metabolites,
Yeah.
not only from the sediment itself, which is becoming anoxic, but from the plant itself,
Yeah.
which is becoming anaerobic.
Mhm.
And for example, the plant root can export ethanol during
Yeah.
the night, but not during the day.
Yeah.
And that will affect the whole microbial system around the roots.
Absolutely.
The plant itself. So it's not, I guess what I'm trying to say is it's not all about oxygen
Yeah.
in this system, and it's not all about oxygen from both the plant and the sediment side.
Yeah.
So I think in designing a study around metabolic activity, you're going to have to consider that the DOC component from the plant itself.
Yeah. Sure.
So
Yeah, and
that's just a comment. John.
No, yeah, that's a really good point, especially when you're thinking about the differences between day and night. Here, I'm more interested in trying to characterize spatial. Like, I don't anticipate doing a lot of these trials at night. It's difficult enough to just see what's going on during the day in these sediments. But yeah, that switch during oxygenated versus or aerobic versus anaerobic is definitely important. Yeah. In terms of both the sediments, but also the plant physiology. I completely agree. I was going to. There we go. How do I do this? Oh, there we go. Okay. Bunch of comments about how they can't hear. Here at the end, okay. Why would you use your DIOL manipulation setup instead of just bubbling with N2 and CO2 to create the? Oh, yeah, that's a really good question. So the reason that I used a sediment-based system is bubbling with N2. It does expel oxygen from the water, but it also affects the PCO2 in ways that are not natural to hypoxia, as you would experience it in situ, right? Because in actual, like in situ conditions, hypoxia is induced by biological activity, right? By the drawdown of oxygen and the production of CO2, which changes the pH. So first of all, it's cheaper to get a bunch of mud from the marsh and put it in a thing instead of to rent tanks of nitrogen. But also it is more realistic to hypoxia, not only in hypoxia is not just low oxygen, it is also change in all of these other biogeochemical parameters that are associated with oxygen. So I thought that was important. So really, I was manipulating oxygen, but I was inducing hypoxia as a way to replicate as accurately as I could manage what was actually happening out in the world. Thank you. Niels.
Yes, can you hear me?
Yes.
Yeah.
Awesome. So I'm glad we could fix the presentation here online because it was really worth it. Thank you so much. And just want to reiterate that I'm really looking forward to working with you. I have one question about the increased oxygen fluxes of oxygen to the sediment with fauna in these low oxygen periods. So you think they are just pumping harder, or what is your suspicion why you see this increased activity?
My suspicion is that they are pumping harder but not longer. So if you look, for example, at so Eric Christensen back in the 80s performed an experiment on nereid worms and how their pumping changed with low oxygen. And the cores that I took had a lot of nereid worms in them, so sort of doing a comparison. And when oxygen got lower, they would regulate their oxygen consumption up to a point, but then after that they would conform. So what I think is happening is that they are maintaining irrigation rates and maybe even increasing to compensate for the low oxygen as the oxygen is declining. is my theory. The problem is that we can't know that that's what they're doing because they were out in the bay in chambers at the time. So that would require doing some more, I think, lab experiments in order to see what behaviorally what they're actually doing when the oxygen gets low. If it's them pumping more intensely, like the amplitude of their irrigation increases, or if they're irrigating for longer, what have you, or if more of them are irrigating, because there was some variability between individuals into how much they pumped when oxygen got low. Like it's not that oxygen goes down and all of a sudden every single layer is like, it varies quite a bit.
Very interesting. I'm looking forward to talking more about these things with you.
Yeah,
Thank you very much.
Other questions?
Well, let's thank Kara. Thank you for coming, right?